Hi, everyone. My name is Amanda Everett, and I'm a PhD candidate at UCSF in the bioinformatics program. So, one of my favorite aspects about being a bioinformatician is that it's actually an incredibly creative job. The data sets we're working with have so much information in them that if you're creative in how you view and ask questions about the data, each time you can find new results and get closer to understanding the full picture. And this is what my thesis project is getting at. So formally, the title is Using Genomic Footprinting to Reveal Cell Type-Specific Transcription Factor Grammar. But conceptually, I'm asking, can we gather more information from our single-cell data about transcription factors? So transcription factors are a class of protein which directly bind to DNA and go on to influence the amount of other proteins in a cell. So we refer to some of them as master regulators of the genome because they interpret the DNA and ultimately drive a whole host of other biological processes. Because they are so critical, you can imagine that if they stop working properly, it can result in many different diseases and disorders. So we need to understand these proteins from a basic biology perspective, but also to drive our understanding of human disease. I believe our best opportunity to understand the fuller picture of transcription factor binding is through a computational method called footprinting. Footprinting is based on a really interesting observation made back in 1978 that if you randomly cut a piece of DNA, you can visually see a gap where transcription factors protect the DNA from being cut. And that's what we refer to as a footprint. In our modern sequencing era, we're actually sequencing those DNA reads and using the computer to search for very localized footprints. This is such a powerful method because with one experiment, you can observe multiple transcription factor binding across the entire genome. So it's one of the more high throughput options available. However, a major limitation of this method is that currently we can only apply this to bulk sequencing. So a large drive of my thesis is to bring these methods into the newest sequencing era of single cell, which would allow for higher granularity. That brings me to my proposal phases and deliverables. So the first big task is to develop and release a simulator that can take real single-cell data sets and generate simulated one based on our variables of interest. Here, like reducing the number of cells. The second task is to apply the footprinting methods to each simulation and calculate how many times it correctly calls a transcription factor binding site to generate a distribution like this. That way we can answer how accurate footprinting methods are for all current and future experiments. I quickly want to add that we're doing this both in a method-specific way, meaning there will actually be multiple distributions, and a transcription factor-specific way. So we'll be able to tell if certain transcription factors, here like the one in purple, have low accuracy overall. Afterwards, there's quite a few paths that open up for this project. The first is using this knowledge to perform footprinting on the wealth of currently available single-cell data sets. So seeking out some of those biological findings that are still left to discover. And the second is to develop a new footprinting method that would increase the accuracy specifically for single-cell data sets. And with that, I'd like to thank the Pollard Lab members and the ARCS Foundation for putting on this event and supporting my education as well. Thank you.